In this video, we will discuss about the programmable surface capsular polysaccharide or the cap of the bacteria for in vivo delivery for therapeutic purpose. So first we will discuss what is this cap. So this bacteria, mainly the Ecoli bacteria, has this natural ability to form this capsular polysaccharide, and this capsular polysaccharide will prevent the bacteria from the host immune evasions or the bacteriophages infections. So what the researchers did is they modified or designed a synthetic cap that can be controlled in a way to either help in the immune evasion, or once their job is done, they can they can be degraded by reducing the cap thickness. So, and these things is depend on the extracellular factors. So if, for example, if they are in the tumors, then they will focus on this immune evasions and helps in the this helps in the delivery of the synthetic circuit that is present within this bacteria. If they are in the normal cells, then they might go towards this clearance. So two of the approaches are mentioned here. First one is enhanced probiotic delivery. So in that case, the cap is the bacteria with the synthetic cap, and it is represented here with uh, thick thickness of the bacteria. And so once they are delivered, they can target the tumors, or those that are not been reached to the tumors, they have the low, their thickness goes down, and this will eventually leads to the bacteria clearance. And one of its how this will work based on the toxicity is shown in the bottom side of the panel B, where we can see that the bacteria with the thick cap has low toxicity, and they will lead to the slow clearance, whereas the bacteria with the thin cap has the high toxicity, and they are prone to high immunogenicity, and they will be cleared in a short duration of time. The other things that can be done is they also designed where the they also designed this cap in a way that it will activate once they are inside the host, or the in situ activations can also be done. In that case, the cap is not already present in the bacteria, but once they are transduced or injected into the host, then if they are in the tumor sites, then they are able to activate these cap formations, and if they are not in the tumor sites, then these cap formations will not be active, and this will lead to the bacteria clearance. So here we can see that with the high cap bacteria are able to translocate to the tumor site, or their translocations is increased, whereas the bacteria with the thin cap is not been able to translocate, and they will lead to the clearance. And these things are being shown in the form of bacterial survivals in the bottom side of the panel C, where we can see that the survival rate of the bacteria with the thin cap is low, whereas survival rate with the bacteria with the thick cap is high. And because of the in situ activations, they are able to see the survival rate of the bacteria from thin cap to the increases as increases with the thickness of the I cap on the bacteria. So once the thickness is increased, the survival probability within the host is also increased. So this is just a overview of what the researchers has developed. We will discuss their results in our subsequent videos, how they have designed their cap, and validate the functionality of their cap synthetic cap design bacteria. So they did their studies or research on the Ecoli bacteria that was discovered in 1917. So I hope you understood their idea and how it can be used for therapeutic purpose. If you need to understand more about their uh, more about their design, or this capsular polysaccharide, synthetic capsular polysaccharide of bacteria, you can go through the reference and how they are controlled with the extracellular environment. You can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.